In 1984, Kerry Mullis developed a new method of amplifying or making many copies of DNA. Although he didn't win a Nobel Prize for his work until 1993, his work revolutionized our ability to research and experiment with genes. His technique was called PCR, or polymerase chain reaction. Today we will be talking about how it works and how it can be used. PCR is based on 3 main stages, and by cycling through these stages, we can take a single copy of DNA and create millions and billions of copies within a few hours. To do PCR, we need 5 main components: a DNA template to make copies of, primers that can bind to a gene of interest, which is the gene we're trying to amplify, nucleotides that can be added to create a new strand of DNA, a heat-stable polymerase such as Taq, and a buffer which maintains sample levels such as pH. The process begins with denaturing at a temperature of approximately 94 degrees Celsius for 1 minute. The temperature alters the shape of the histone proteins that DNA is wrapped around, which allows the DNA to unwind and makes it more accessible. The temperature also breaks the hydrogen bonds that hold the 2 strands together, allowing them to separate. The next step is annealing, which occurs at a temperature of approximately 55 degrees Celsius for 45 seconds. The cooling of the temperature allows our primers to bind to the DNA. Primers are short sections of DNA that have a sequence of nucleotides that is created specifically to bind to the edges of a certain gene. The last step is extending, which happens at approximately 72 degrees Celsius and is done for about 2 minutes per 1,000 base pairs. This stage is when the polymerases add nucleotides off the 3' ends of the primers, extending the new DNA strand. The nucleotides are added in an A with T and G with C pattern, so the old and new strands are complementary. To help with the process of heating and cooling the DNA samples efficiently, we usually use a thermal cycler, which can be set to certain temperatures and times based on the samples. The series of 3 stages is repeated 30 to 40 times in the cycler to create enough copies of DNA. At this point, you might be asking, what can we do with so many copies of the DNA? Well, there are many applications of PCR in forensics, genetics, and even the detection of pathogens. Many people may now be familiar with real-time or RT-PCR COVID tests. During RT-PCR, fluorescently tagged markers are added into the sample, which will glow when added to the new growing strand of DNA. If the viral RNA is present in the sample, the amount of fluorescence will increase as more copies of the DNA are made. The fluorescence can be tracked in real time, and the whole process can be completed within a few hours. This makes it much quicker than most other types of PCR, and is why it's so widely used to test for COVID infections. As we learned today, PCR is a relatively simple lab procedure that can be done quickly and has many uses. It will be a vital tool as we continue to study the human genome and work to prevent the spread of diseases such as COVID-19. I hope you learned something new and that you have a new appreciation for the COVID test you might be taking. Thank you especially to my amazing medical interventions teacher, Mr. Shokin, who taught me all about PCR.